Dear colleagues, dear friends, it's with great pleasure that I'm going to introduce to you the next session. This is the second day of the TIFF meeting, and it's the evening session which is entitled Innovation in Hemoglobin Disorders. I'm Antonis Katamis from the University of Athens, and I will chair with this session along with Professor Capellini, who is replacing Professor Taher, who's unfortunately had a lot of difficulties connecting, so he's not going to be able to be present for this session. We have heard a lot of very interesting presentations, especially during the current strategies on thalassemia, and now we're going to move to the future, what to expect. And I think this is a very interesting session that you're going to hear. We're going to talk about the innovation in hemoglobin disorders. I think this is the future, how the field will change over the next years. And I'm very privileged to introduce the first speaker. Stefano Rivella is a very well-known figure on the thalassemia research field. He's a professor of pediatrics at the Children's Hospital of Philadelphia at the University of Pennsylvania, and Kof, as we know, Kwame Ohim-Frimpong, chair of sickle cell anemia at the same university. He will talk about the authorized and ongoing clinical trials of medical products and devices for the treatment of thalassemia.
First, I would like to thank the organizer for inviting me to discuss this important topic. So these are my disclosure. Also want to mention that none of the data that we present today were supported by any of the companies listed on this slide. So let me start mentioning very briefly that beta thalassemia, as we know, is a congenital disease where the mutation lead to an excess of alpha chains that form hemicrons, which are toxic aggregates that impair the quality of the RBC and lead to the reduced lifespan of these the erythrocytes in circulation. This leads eventually to ineffective erythropoiesis and a reduced proportion of erythrocytes in circulation. The novel therapies that we'll discuss today are focusing on the improvement of the RBC quality, such that they can lead to improvement of ineffective erythropoiesis, and also the use of therapeutics that can improve ineffective erythropoiesis, or strategies that lead to the synthesis of therapeutic hemoglobin, such as adult or fetal hemoglobin. However, during my talk, I will not discuss drugs such as pyruvate kinase agonists or activators of fetal hemoglobin, which are being covered by other talks. So let's start with focusing on drugs that improve RBC quality by iron restriction, which also here are defined as iron restriction drugs, or IRD. So what's the main mechanism? So the IRD drugs basically limit iron absorption, dietary iron absorption, leading to decreased transferrin saturation. As a consequence, there is less iron going to the erythroid cells and a reduction in heme synthesis. As we've been shown in several publications by my group and other groups, eventually in thalassemia, this leads to a decrease in formation of hemicrons that lead to a better quality and an increased amount of red cells in circulation, because now the red cells live longer and accumulate in the periphery. Some of the IRD molecules that are being developed right now are hepcidin agonists or activators, ferroport inhibitors, and transferrin molecules. And all these drugs basically have the property to reduce iron absorption or to decrease transferrin saturation. So these are basically some of the drugs that are now presently used in clinical trial, listed here below. Some of them are also called tempura 6 inhibitors, which are basically drugs that increase hepcidin expression, which is the hormone that controls iron absorption. And these basically can lead to decreased iron intake and again, decreased transferrin saturation. Another trial is based on administration of apotransferrin, so transferrin without iron. And the goal again is to decrease transferrin saturation. So these clinical trials listed here are ongoing right now, and we hope to see if the results of this new application will be positive in coming years. So the next set of drugs I will briefly discuss today are drugs that improve ineffective erythropoiesis, also labeled here, I decided to call them ineffective erythropoiesis modulating drugs, or IEMD. And basically the mechanism is depicted in this slide. So if we consider this normal or steady state erythropoiesis in let's say wild type, in a wild type or normal condition, we see that there is a certain proportion between the progenitor erythroid cells, the cells that will make eventually erythrocytes, and a certain amount of erythrocytes in circulation, which is more abundant than number of progenitor cells in the bone marrow. In thalassemia, we have a complete reverted relationship where there are more progenitor erythroid cells in the bone marrow, and because this erythropoiesis is ineffective, there is, despite the larger amount of progenitor, there is a reduced number of erythrocytes in circulation. With drugs such as, that we'll discuss in the next slide, such as luspatercept, for instance, which is the prototype of drugs that control or modulate ineffective erythropoiesis, the goal is basically to reduce the proportion of progenitor erythroid cells, but allowing them to be more effective in producing erythrocytes in circulation, mature red cells in circulation, such that now this proportion between progenitor and mature red cells is more similar to what we observe in normal individuals. So the prototype of the drug that allows to do this is called luspatercept. This is a very important paper published by Dr. Cappellini as the first author, that show the use of this drug has been able and significantly increase the red cell production in patients with thalassemia, or decreasing their requirement for blood transfusion, and additional positive effects that are listed in this manuscript. Right now, this drug is already been approved, available to patients affected by beta thalassemia, but there is an ongoing clinical trial to see if this drug is also effective and safe in pediatric patients. So I would like to conclude this portion of my talk on IRD and IEMD drugs with some consideration. So first of all, I'd like to mention that the mechanisms of action of IRD and IEMD are not completely characterized. I think more work is required to fully understand their potential, but also potential limitation. For instance, IRD drugs, iron-restricted drugs, improve not only RBC production, but also ineffective erythropoiesis. Drugs that modulate ineffective erythropoiesis improve RBC quality. So the point is, are there additional mechanisms of this drug that we have not been yet able to elucidate? In addition, I also want to point out that it's possible that both this group of drugs and perhaps pyruvate kinase agonists that we described in other talks could be combined, leading to a positive additive effect on RBC production, ineffective erythropoiesis, iron overload, and anemia. In addition, I also wanted to consider that these drugs may also have a role together with gene therapy application, such as in improving the quality of the bone marrow microenvironment and iron overload before bone marrow transplant, and potentially also helping those patients that didn't achieve optimal hemoglobin production after gene therapy. In the second part of my talk, I'm going to discuss the more gene therapy application and therapies and technologies for the treatment of beta thalassemia. I will start with gene addition. So gene addition is basically a mechanism that is functional to increase or to deliver a functional copy of the beta globin gene into the cells of the patient, where obviously the beta globin gene is mutated or not functional. So the advantages of these technologies are basically delivered right now of utilize basically lentiviral vector, which are viral vectors that are able to, again, as I mentioned, to deliver a corrected copy of the beta globin gene to the patient cells. The advantages of these approaches are this technology is independent from the mutation of each patient, and eventually can lead to overexpression if the expression of the therapeutic gene is required in superphysiological doses. The disadvantages are obviously random integration. This approach doesn't allow the transgene to be integrated in specific region of the genome of the patients. Some of the vectors being utilized so far may require high vector copy number to reach therapeutic levels of the therapeutic proteins such as beta globin or gamma globin. And these obviously can lead increasing chances of genotoxicity, as random integration and high vector copy number can eventually lead to integration close to, let's say, a proto-oncogene, activating its function and leading eventually to leukemia. I just want to mention that in the last year, a few cases of MDS AML were observed in patients with sickle cell anemia that were treated with a vector from the Bluebird company that also utilized sortation with beta thalassemia. But subsequent studies indicated that the vector insertion did not contribute to the leukomegenesis, and that also typical AML-related mutations were identified in these patients, which were independent by vector integration. And so the point and the studies now undergoing are trying to understand if these cases of clonal hematopoiesis, MDS AML, may be limited to sickle cell disease, and if this is maybe related again to sickle cell disease because it's a disease where the chronic inflammation may increase the chances of clonal hematopoiesis, and together with this kind of treatment can lead or exacerbate a phenomenon that may be already present in patients with sickle cell anemia. So in this slide, I want to show two of the, I think, the most relevant paper that discuss or that were fundamental to lead to clinical trials. On the left, you can see the paper published by Dr. Cavazzana that basically indicated that a vector generated by the group of Bluebird was able to increase hemoglobin levels in patients affected by beta thalassemia. Similar endpoints were also achieved by a vector generated by the group of Dr. Giuliano Ferrari in Milan. And below you can see ongoing clinical trial, many ongoing clinical trial sponsored by Bluebird. And I also want to mention that in addition to these major vectors, there are additional trials that have been undergoing right now in China, although there is limited information about the structure of the vector that being utilized in these trials. So now, before I move to the additional gene therapy approaches related to genome editing, I also wanted to introduce briefly the target of these approaches, which is basically reactivation of fetal hemoglobin. So as we all know, before birth, we express a different kind of hemoglobin called fetal hemoglobin. And after birth, and this is the fetal hemoglobin is basically produced by activation or expression of a gene called the gamma globin gene. After birth, the gamma globin gene is switched off, while the beta globin gene is switched on, and this beta globin gene is responsible for production of so-called adult hemoglobin. However, in patients affected by, for instance, beta thalassemia, while they are born with normal level of hemoglobin because the hemoglobin is basically produced by an active and functional gamma globin gene, after birth, as the gamma globin gene decreases in its expression, the mutant beta globin gene takes over, and now we have clearly observed clinical observation of the disease. So it's been shown in the last few years that one of the master regulator for the switching between fetal and adult hemoglobin is a gene called BCL11A, and also been shown that if we inactivate this gene in erythroid cells, we can reverse the switching such that the beta globin gene will be expressed at a lower level and the gamma globin gene at a high level. So in other words, if we can target the gene called BCL11A, we can theoretically reactivate expression and synthesis of fetal hemoglobin and decrease the expression of the mutant beta globin gene. And this has been the target of a technology called genome editing. In particular, what I define a genome editing application to gene disruption. So this is basically a system where a variety of tools called indicated as a CRISPR, zinc finger nucleases, talents, etc., can basically deliver inside the cells a system to target and destroy a specific DNA sequence. Here, for simplicity, I'm using the CRISPR Cas9 approach, where basically these two elements, a Cas9 protein and a guided RNA, are utilized together to identify a sequence in the genome that can be destroyed and activated. In this case, the sequence of the BCL11A gene. As in achieving this result, again, the BCL11A gene is suppressed, and now we can reactivate fetal hemoglobin. The advantage of this approach is that gene disruption is very efficient, and but there are also disadvantages, such as these in some publication. It's been seen that gene targeting can lead to genome instability, and also that homologous that this kind of approach may require low level P53 gene that can lead to clonal selection. So in any case, this approach has been utilized by a company called Vertex. And when the first paper and the first patient treated with this approach, in which the BCL11 gene was targeted by this approach, was were shown in this paper, and the results were very positive, such that the patient now express high level of fetal hemoglobin and are transfusion independent. Obviously, this trial is still ongoing, and we are obviously looking forward to see more patients treated with this technology. But what are basically potentially desired improvements that I listed here in this slide? On one side, I think genome editing can be improved in targeting quiescent cell, because this is a limitation with this approach. Obviously, we need to be sure that there are not potential genome toxicity effects related to this technology. The only time will tell us. And obviously, I would rather correct mutation than destroy genes. For the gene addition, I think maybe more powerful vectors are required to increase the ratio between the curative hemoglobin and the number of integration. And it would be optimal also to achieve use a system where there's no random integration. And I think in either way, for gene addition and genome editing, I think it would be important to reduce or prevent the toxic effect of myeloablation in patients, and also to reduce the manipulation of hematopoietic stem cells or increase the hematopoietic stem cell repertoire. And I think that I would say what I call the holy grail of gene therapy is really to target this mutation in vivo by an in vivo gene delivery approach. So from the perspective of achieving mutation correction rather than gene disruption, there are already tools available. For instance, this is an approach called base editing, where the Cas9 protein is being modified in order to create only a single single strand cut in the DNA, followed by a precise modification of a base such that the mutation can be corrected. This approach is not yet in clinical trial, but I know by just looking at publications and reading news that a variety of companies are now very interested in developing this technology, such that in the future we may be able to correct directly mutation in the patient cells, rather than disrupting gene or adding genes in random integration. So from my perspective, what we're trying to do in my lab is actually increasing the power of lentiviral vector technology. These slides basically summarizes a recent paper we published in my laboratory, where basically we generated a variety of novel lentiviral vector in which the beta globin gene was expressed using a variety of novel different genomic elements to increase the expression of the beta globin gene. And while we were identifying the best vector for expression of the beta globin gene, we also compare the expression of this vector to the vectors that are currently in clinical trial or approved for treatment of beta thalassemia, such as the vector BB305 by Bluebird. And basically using a variety of approaches, basically using cells in culture, patient-derived cells, and animals affected by beta thalassemia, we identify a novel vector called LS20, which is able to express between 40% and 150% more hemoglobin than vectors that are currently in clinical trial or utilized for the treatment of beta thalassemia. And now I want to basically almost end my presentation, also providing what I think is basically the future of gene therapy. I think that the improved technology that I expect people to develop in the next few years are basically aiming to the elimination of random integration and avoiding any genome editing on target effects, and therefore improving gene addition, gene editing techniques. Another desirable improvement is the reduction of elimination of toxic myeloablation, which we know now it's a requirement for gene therapy, but also comes with a variety of side effects, including lack of fertility and potential development of MDS in the long term. And of course, as I mentioned before, ideally, we would like to create a system where we can target the hematopoietic stem cells directly in vivo, and this will basically overcome completely the requirement for myeloablation. And I want to finish just because, as you know, in my title, there is also the word device. And for that reason, I check among all the clinical trials if there was something novel that could be also help patients affected by beta thalassemia. And I discovered that there is now a device, a microneedle array patch that's being developed to deliver a local anesthetic in order to reduce the pain in pediatric patients that require blood transfusion. And I think also with these tools, we can hopefully improve the quality of life of patients affected by beta thalassemia. And my last slide is basically just the slide to thank most of the people in my laboratory that help me every day with our work on iron metabolism and gene therapy. And with this, I will thank you, and I'm happy to take any question. Thank you very much.
We thank Dr. Rivella for this nice presentation. We will leave it at the end for discussion. Now is my great pleasure to introduce Perla Eleftheriou. Before doing that, let me remind you that all the participants, all the attendees may send questions through the chat, and we will take care at the end of the session. Once more, I present you the apologies of Dr. Taher, who is not able to be connected. Dr. Eleftheriou is very well known. She already had some presentation in the previous session, and now the title of her presentation is Authorized and Ongoing Clinical Trials of Medical Products and Devices for the Treatment of Sickle Cell Disease. Perla is a consultant hematologist at the University College of London. Please, Perla.
Good afternoon, everyone, and thank you very much for asking me to talk about novel treatments and clinical trials in sickle cell disease. I will start sharing my slides. So I'm Perla Eleftheriou, hematology consultant at University College Hospital in London, in the UK. And I will be talking to you about the most important news from clinical trial data in the last decade. But I will start with reminding us about the most important key aspects in the pathophysiology of this disease. So sickle cell disease is a progressive and multi-system disease which can lead to many complications early on in life and as the time passes. There is significant heterogeneity of the clinical phenotype, but complications accumulate with the time and reduce quality of life and life expectancy. This disease has a huge impact on quality of life. 1/3 of patients report pain every day. The overall quality of life is poorer than even this of patients with, for example, cystic fibrosis or cancer treatment, cancer treated with chemotherapy. And sickle cell disease patients with end-stage renal failure or end-stage renal disease have even more reduced quality of life. The sickle hemoglobin polymerizes in the red cell when deoxygenated, and this leads to sickling of the red cell and hemolysis. Phosphatidylserine exposure plays a role in the sickle cell red cell adhesion properties and also promotes activation of coagulation. Adhesive interactions between sickle red cells, neutrophils, platelets, and endothelial cells are increased, which also leads to activation of inflammatory pathways and further activation of the coagulation cascade. Now, following deoxygenation, the mutated hemoglobin, so the hemoglobin S molecules, polymerize to form bundles, and this results in the red blood cell sickling and consequently impaired blood flow. The sickle hemoglobin polymers have a reduced P54 oxygen, reducing oxygen carrying capacity, and the right-shifted oxyhemoglobin dissociation curve of sickle cell disease results in abnormally low arterial oxygen saturation, even when the oxygen partial pressure is normal. Free heme and hemoglobin and increased radical oxygen species levels cause abnormally increased endothelial expression of addition molecules such as E-selectin and P-selectin, and this leads to recruitment of neutrophils which trap red blood cells, causing further vasoocclusion. And this is exacerbated by the dense dehydrated sickled red cells. Principles of management: prevention of complications is absolutely key. Appropriate treatment of acute and chronic complications, rapid access to care for acute complications with specialist oversight, early and appropriate introduction of disease-modifying treatment, effective anticipation and management of psychosocial difficulties, and all this in close cooperation between community-based and hospital-based care teams. So this busy slide, yet very important, illustrates how throughout the years from 1910 to now, 2021, there has been a remarkable progress in the insight gained into sickle cell disease, as well as in the development of new treatments. However, you can see that the progress was extremely slow at the start, with very little coming forward altogether in a whole century compared to what has been happening in the last decade. You see that from 1910, when the first case report came through, until in 1980s, there was pretty much nothing moving in sickle cell disease. And in 1980s, we see the value of prophylactic penicillin being recognized, the trials with hydroxyurea and other hemoglobin F inducing agents starting. And in 1984, we had the first allogeneic stem cell transplant in sickle cell disease. Important milestones to note, looking at this timeline illustration: 1995, the big MSH study of hydroxyurea was published. 1990 to 2000, hydroxyurea FDA was approved. So FDA approval of hydroxyurea. Trials in the use of blood transfusion in sickle cell disease progressed. 2006, gene therapy vector development. 2015, gene therapy advanced further. 2016, we see crizanlizumab coming up in the form of the SUSTAIN study, the GBT studies, and other important ones. 2017, FDA approves L-glutamine. And in 2019, more than 700 clinical trials are registered in sickle cell disease. 130 studies in relation to stem cell transplantation in sickle cell disease, and crizanlizumab is FDA approved, voxelotor is FDA approved, all in 2019. So standard of care, let's talk about that, what we have at the moment in our hands to treat the patients outside clinical trials. Hydroxycarbamide, blood transfusions, bone marrow transplantation. Hydroxycarbamide, multiple beneficial physiological effects. We know this through the studies and through observational studies and case reports. It reduces the vasoocclusion in terms of frequency and intensity, the acute chest syndrome, hospitalizations and mortality. It's usually the first-line treatment for disease modification. Going on to blood transfusions, usually given as long-term exchange transfusions as preventative treatments. They reduce the sickle hemoglobin percentage, endothelial injury and inflammatory damage, but the downside is the alloimmunization risk, iron overload, and viral infections, the latter being a very small risk indeed. Bone marrow transplantation is a curative treatment, of course, given or applied in the mostly in the form of an HLA-matched sibling allogeneic transplantation, although new insights have been gained into the haploidentical stem cell transplantation. So again, going into a little bit more detail about the standard of care, hydroxyurea, fetal hemoglobin inducer, it significantly reduces the incidence of VOCs, the hospitalizations and mortality. Downsides, myelosuppression, so it's a dose-limiting effect, reversible on cessation of treatment, affects less than 10% of patients under the maximum tolerable dose, and affects about 50% of patients who reached maximum tolerable dose. Significant degree of heterogeneity in animal studies, and some other side effects which might interfere with the compliance of the patients with hydroxyurea, such as gastrointestinal disturbances, skin and nail, hypo or hyperpigmentation, thin hair. Blood transfusions. So there is evidence for stroke prophylaxis, so primary and secondary stroke prophylaxis, absolutely, and is also applied as a second line of treatment to prevent frequent and intense vaso-occlusive crisis and acute chest syndromes. The prevention of other complications outside stroke really remains pragmatic, based on observations and clinical experience, and a lot of this information that we get to justify using regular blood transfusions to prevent VOCs and acute chest syndromes had been extrapolated from the randomized trials in the pediatric stroke stroke prevention. So the aim of the blood transfusions is to reduce the sickle hemoglobin to less than 30% for primary or secondary stroke prevention. And for other indications, there's no evidence for any particular range of the sickle hemoglobin, but we tend to keep it between 30 and 35%. And this is where we see the benefit of transfusions when we keep this the sickle hemoglobin in this range. So indeed, transfusion ameliorates severe disease. You can see this in this Wyszynski study, which was published a couple of decades ago. So definitely reduces hospitalizations, reduces acute chest syndromes, viral hospitalizations per year, bacterial infections hospitalizations per year. Allogeneic hematopoietic stem cell transplantation. First transplant for sickle cell disease in 1984. Overall, only 3,000 patients actually with sickle cell disease have undergone this treatment worldwide. 14% of sickle cell patients have a healthy matched related donor, but you can see one of the reasons this procedure or this intervention cannot be applied more widely is because of the very low proportion of our patients actually having a matched related donor. Graft failure in the matched transplant, 5 to 10%, in the mismatched 13 to 17%, and graft versus host disease increases with age in the less than 16 years of age, acute GVHD about 13%, chronic GVHD about 15%, and this goes up slightly when the age increases to above 16 years of age. But generally speaking, the sibling matched allogeneic stem cell transplantation in selected sickle cell patients is a very promising intervention for certain patients. So this paper was published in Blood in 2017. It was an international survey of all the results of HLA identical sibling stem cell transplantation collected between 1986 and 2013. Involved 23 countries and more than 100 centers, 1,000 patients, 154 were adults, 5-year overall survival, 95% in those who were transplanted less than 16 years of age, and 81% in those transplanted older than 16 years of age. Indications for considering transplantation in patients with sickle cell disease would be severe sickle cell related end organ damage, such as stroke or abnormal intracranial vessel velocities, renal damage, liver disease, or modifiable complications not improving by hydroxyurea or blood transfusions. For example, despite sickle modifying treatments, increased hospitalizations with VOCs or acute chest syndromes. And this is the paper which was published in 2019, looking at the haploidentical approaches in those patients with no fully matched related donor. And this essentially supports the idea that haploidentical approach is or should be considered within, at the moment in the UK, it's within a clinical trial, which is hopefully going to open up very soon. So this illustration gives you an idea of how, when we talk about standard of care medications, but also looking at how we can work on new innovative treatments, where to target the different steps in the pathophysiology of sickle cell disease. So, for example, we can change the genotype, and this is the rationale behind the allogeneic stem cell transplantation, as well as the gene therapy. We can target the sickle hemoglobin polymerization, and this is a rationale between the fetal hemoglobin, the rationale of the fetal hemoglobin inducers, for example, or the drugs which affect the hemoglobin oxygen affinity. We can target the vaso-occlusion by inhibiting adhesive interactions between cells and endothelium, and we can target inflammation. And for example, that's one of the L-glutamine functions. So novel drugs for sickle cell disease can be fetal hemoglobin inducers, they can be cell adhesion inhibitors, they can be anti-sickling agents, and endothelial cell dysfunction reducing agents. So this table shows you timelines of all important, but not all of them, of course. This is not an exhaustive table, but it gives you a timeline of a few of the most important milestones and products and treatments that we have seen coming through in the last decade. So you see hydroxyurea, standard of care, L-glutamine targeting vaso-occlusion by increasing NAD and NADH and decreasing adhesion, FDA approved in July 2017. The P-selectin inhibitor crizanlizumab, FDA approved in 2019, and NICE approved in the UK November 2021 in a managed access agreement scheme. The voxelotor produced by GBT, FDA approved in November 2019. It targets hemoglobin S polymerization. And you see that the HLA matched transplant is standard of care, but the haploidentical transplant is in phase 3 trials. And other products rather here to note: the Imara 687 hemoglobin F inducer phosphodiesterase inhibitor in phase 2 trials. We have the gene therapy treatments, which are in phase 2 clinical trials. Some of them are in earlier than phase 2 clinical trials. And next, to talk a little bit, just a little bit more about L-glutamine, or more about specific, rather, IMPs and where we are with them and what the rationale is. So L-glutamine is a precursor of arginine, which is a precursor of nitric oxide. And this phase 3 trial shows promising results, some promising results at least. For example, the time to the first and to the second sickle cell crisis is prolonged compared to the placebo arm, and the overall crises were less, from an average of 4 down to 3 per year. There are some downsides. For example, it comes through, so it comes in a powder form to be dissolved in water or juice twice a day, which is challenging for patients. And also there's no laboratory way to monitor the response or the compliance of the glutamine. Voxelotor, it's a once daily oral treatment which binds to the hemoglobin and stabilizes the molecule in the oxyhemoglobin, so in the R state, and therefore safely increases the oxygen affinity to create a fraction of non-sickle hemoglobin, which then inhibits the sickle hemoglobin polymerization. And the data we have for the Voxelotor efficacy and safety come through the phase 3 randomized double-blinded HOPE study, which the primary endpoint of which was the hemoglobin response, and secondary endpoints were reduction of change of hemolytic markers and annualized incidence of VOCs. So the primary endpoint was met, as more than 50% of patients at the 1500 milligram dose achieved more than 1 gram per deciliter hemoglobin increased, and significant reduction in the markers of hemolysis at week 24 was seen. And what did we learn from Voxelotor is that by blocking sickling of 26.5 of roughly 30% of the sickle hemoglobin, we can increase the hemoglobin by more than 10 grams per liter in 7 to 2 weeks in 90% of the patients. And there was some suggestion of perhaps decreased rate of VOCs per year. Crizanlizumab sustain trial data. So this is an anti-P selecting monoclonal antibody, which is given in the form of an intravenous infusion every 4 weeks. So the sustain trial showed a reduction of VOCs by 45% on the high 5 milligram per kilogram dose, longer time to the first VOC and longer time to second VOC, and no change in the hemolytic markers. Imara 687 selective phosphodiesterase 9 inhibitor, which increases the fetal hemoglobin. It actively hydrolyzes cyclical GMP to GMP, and therefore the increased cyclical GMP levels reactivate fetal hemoglobin. Currently, we are in phase 2B study in relation to sickle cell disease, looking at this IMP. The duration of the study is 1 year. The primary endpoint is more than 3%, 3 or more than 3% increase in the fetal hemoglobin versus placebo. And also clinical endpoints would be the VOC rate, acute chest syndrome rate, and other clinical markers. So that is still data is still pending from this, and there has been there has been and there is still interest in more fetal hemoglobin inducers, decitabine, for example, which is a hypomethylating agent. There's ongoing trials looking at this. Panobinostat, although now is not recruiting this study. Metformin with hydroxyurea. We talked about the Imara. So there is a lot of interest in looking at the at how we can increase the fetal hemoglobin using or targeting different different steps in the in pathophysiology to achieve this. So here I'm going to highlight the pyruvate kinase activation as a potential treatment of sickle cell disease, as it was not exactly really the aim when these agents were first developed. And it's recognized that the pyruvate kinase activity is decreased in the sickle red cell, and therefore less ATP is made, and hence less defense against oxidative stress, which also lowers the oxygen affinity of the hemoglobin. And this is a phase 1 and multiple ascending dose study looking at the safety and pharmacokinetics and pharmacodynamics of a particular PK activator in healthy and sickle cell disease subjects, which is the results of which are awaited. But there are 3 currently PK activators, as far as I know, undergoing clinical trials. So there is some promising development here. Crovalimumab, some agents can reduce endothelial dysfunction, which is responsible for long-term effects in sickle cell disease. And we know complement system is activated in sickle cell disease, and hence there is a lot of interest, generally speaking, in complement inhibitors. And this is one of those that is now in phase 1b studies. Another agent which might be reducing the endothelial dysfunction in a sort of nitric oxide related mechanism, the arginine, and it's been looked at in the form of IV infusions in a phase 1-2 trials in the States. And although there is a lot more than what I discussed going on, this illustration summarizes the most important current and novel different interventions and how they act at different pathobiology targets. And also to note the Hemopexin phase 1 clinical trial, which is currently open at multiple sites, and the expectation is for IV Hemopexin to reduce the endothelial dysfunction. But yes, it remains unknown if VOC or endocrine damage will be prevented by doing this. The last few slides on gene therapy. Gene therapy is regarded as the next big promise, of course, in terms of curing the disease, and there is an increasing number of ways one can apply gene therapy in this disease. You can add an anticycline or just a different hemoglobin A hemoglobin, or you can correct the mutation, or you can interfere with the fetal hemoglobin induction gene. And it remains uncertain, though, if gene therapy will actually cure sickle cell disease or lead to a milder form of sickle cell disease. And this, so the CRISPR study looked at the infusion of this product, which increases the fetal hemoglobin levels in erythroid cells in vivo. And this was presented. So the CLIMB sickle cell disease 1-to-1 studies of CRISPR modified CD34 hematopoietic stem cell, stem and progenitor cells. The results of this study was published in ASH 2020, and essentially it shows that the proportion of, the primary endpoint was the proportion of patients with fetal hemoglobin more than 20% sustained for at least 3 months after starting, for at least 3 months, starting 6 months after the product infusion. And the safety data, which was presented at ASH, showed actually that it was a well-tolerated treatment overall. The AEs, the adverse events, were generally consistent with myeloablation and autologous stem cell transplant. There was nothing unexpected and nothing related to the actual infused product. And to conclude the data which was shown from this study, the clinical proof of concept for this product had been demonstrated, and clinically meaningful fetal hemoglobin and total hemoglobin levels were observed early and maintained across all patients who received this treatment. So it is very important to weigh up, when we discuss allogeneic stem cell transplantation and gene therapy, is very important to weigh up risks and benefits, especially if they could be eligible for both. And of course, if we break down the pros and cons of each intervention for the allogeneic stem cell transplantation, we have much more data. We know essentially more about it. We can achieve 100% chimerism and therefore achieve complete cure. Whereas with the gene therapy, the outcome could be a milder form of sickle cell disease than a complete cure. And there is some concern about some off-target effects, for example, myeloid neoplasms, but this remains to be answered. And when it comes to other problems with allogeneic stem cell transplantation, GVHD, immunosuppression, long-term effects of radiation. So those are all aspects that we need to know well in advance, in depth, and be able to discuss and weigh up with the patient. And this takes a lot of consultations usually, and the input from family and support from family as well. So the risk of development of long-term complications is high and the median survival is decreased. So it is obvious that early intervention is key. However, the risks and benefits of the different options need to be carefully weighed up with the patient in the center and ideally supported not only by the healthcare professionals looking after them, but by family as well and friends. And the option of best supported care, curative and clinical trial options need to be very thoroughly discussed with the patient. So to conclude, regular comprehensive reviews and monitoring for early signs of organ damage is absolutely key. We should have lower thresholds for the use of hydroxyurea and blood transfusions as preventative measures for end organ disease. We should be able to manage aggressively treatable comorbidities and acute deterioration. Primary goal in management, especially of the older adult, is improving the anemia and hemolysis and minimizing organ damage. We should be more proactive in considering curative treatments previously offered only to the younger patients. And curative or experimental interventions should be discussed early on before complications render the patients ineligible for this treatment. Thank you very much for listening to me.
Antonio, you're mute. Okay, maybe I can introduce the next speaker. Yeah. In the meantime that Dr. Kattanis reestablish the microphone, I may introduce Dr. Campion from Silence, who is giving us a talk on exploring the potential of gene silencing in thalassemia. Please, Dr. Campion.
Many thanks for inviting us to present today. As a company that is committed to researching a potential treatment for people with thalassemia, it has been a great pleasure to collaborate with the Thalassemia International Federation on a number of different projects in the past couple of years, from raising awareness of the condition for International Thalassemia Day to gaining the advice of people living with thalassemia to inform our clinical trials. My name is Giles Campion, and I'm Head of Research and Development and Chief Medical Officer at Silence Therapeutics, a biotechnology company working to make the best use of gene silencing technology for new medicines for a wide variety of diseases caused by genetic factors, technology that we would like to explore with you today.
And my name is Alberto Martinez, Senior Director for the Clinical Science team, leading on the development of our potential gene silencing medicine candidate. which we hope will ultimately improve the lives of people with thalassemia.
Over the course of the next 20 minutes, we are going to explain what exactly is gene silencing or RNA interference, which is the biological process we're focusing on in our research and its potential use for people with thalassemia. Alberto is also going to share more about our research program in thalassemia and our preclinical and clinical findings to date. Science Therapeutics is a biotechnology company with more than 2 decades of research behind it in the field of gene silencing. We are working to make the best use of RNA interference technology for new medicines for a wide variety of diseases caused by genetic factors. Our vision is clear: people's lives around the world by silencing diseases through our precision engineered medicines and driving positive change for the communities around us. We are currently focusing on 2 key areas of unmet need: iron loading disorders like thalassemia and myelodysplastic syndrome, and cardiovascular disease caused by underlying genetic factors such as high Lp(a). Our research and development facilities are in Berlin, with headquarters in London and a US office in New Jersey. Gene silencing is a relatively new technique that makes use of the body's natural processes to control disease by suppressing or silencing specific genes that are associated with certain diseases. In this context, silencing means temporarily blocking a specific gene's message that would otherwise trigger an unwanted effect. Gene silencing is a fast-moving area of research. It has the potential to provide highly targeted treatments for specific genetic diseases as a potential lifelong treatment and has already been approved for use in some countries. There are a number of different types of gene silencing methods. At Silence Therapeutics, we're focusing on a biological process called RNA interference, or RNAi for short. RNAi is a feature that all our bodies are born with and is designed to control the activity of the genes or defend the body against viruses. This is the mechanism we'll be explaining today. If you have questions about the other methods, please feel free to send them via the meeting organizers, and we would be happy to explain some of these different approaches. For all the methods, it's important to note that gene silencing is different from gene therapy. Gene therapy involves introducing new DNA into the body to permanently correct a missing or faulty gene. Gene silencing has a controllable, reversible effect, which offers advantages if there are side effects. It is also highly specific and targeted, which means that the results are predictable. So what is RNAi specifically? DNA contains the genetic code of organisms. It has all the instructions that a living organism needs to grow, reproduce, and function. This information is contained in 2 chains that curl around each other to form a twisted ladder structure called a double helix. DNA remains confined to the nucleus of the cell, and it relies on another molecule, RNA, to deliver its instructions and translate them into action. RNA is physically different from DNA. It contains only one single chain and is chemically more reactive than DNA. Unlike DNA, which is unable to leave the nucleus, RNA is more mobile, and there are several different subtypes of RNA which perform different functions. RNA molecules read the instructions provided by DNA, take a copy, and activate the processes required to make them happen. When the instructions are turned into a functional product, such as a protein, this is called gene expression. Essentially, RNA molecules use the blueprint provided by the DNA to instruct the amino acids on how to make the specific protein formation. These instructions are carried to other parts of the cell for processing by a type of RNA called messenger RNA. It is messenger RNA that tells the cell which building blocks it should use to create the protein. In certain diseases, the DNA is mutated or abnormally expressed, and messenger RNA is created from these mutated genes. In some cases, mutations instruct the cell to produce too much protein, or the protein does not work, triggering an unwanted effect and or causing disease. RNAi was a breakthrough discovery in the field of molecular biology in the late 1990s. The scientist who discovered it received the Nobel Prize in 2006. It is a control system that occurs naturally as part of the immune response. The immune response is the body's defensive reaction to invaders like viruses, bacteria, and parasites. Put simply, RNAi molecules clear up mutations, correct mistakes, and attack viruses that have infiltrated DNA. They are involved in the production of new proteins, but they can also work in reverse, moderating or adjusting the activity of the genes when needed. They reduce or block gene expression by neutralizing targeted messenger RNA molecules and decreasing their activity. This is RNAi in action. During RNAi, strands of RNA are divided or diced into small pieces which are called small interfering RNAs. These attach to mRNA targets and block their action, triggering the process of RNAi. This has the effect of turning down or silencing the production of the disease-associated protein, allowing the cell to revert to its normal healthy state. While RNAi is a natural biological process, it can also be deliberately triggered. The idea behind gene silencing is to intervene in the process of gene expression before the protein is made. Introducing an anticode sequence to block problematic mRNA triggers the RNAi process, making it possible to silence or temporarily switch off specific disease-associated genes. Gene silencing molecules are designed to mimic siRNA, allowing them to attach to specific parts of the gene in the same way, therefore blocking the gene's message from being delivered. The key challenge is to deliver siRNA mimicking molecules to the target cells in an efficient and safe way. By temporarily blocking a specific gene's message, gene silencing molecules halt or reverse the process of disease by targeting the underlying disease mechanism rather than the symptoms it causes. I'd now like to hand over to Alberto to discuss our approach to engineering these gene silencing molecules to control disease.
Unlike small molecules and antibodies, it is possible to engineer siRNAs to silence any gene involved in the cause or pathway of disease by blocking the production of disease-associated proteins. As Giles mentioned earlier, use of a tailored and precise approach to silencing targeted genes in specific parts of the body leads to a more predictable treatment effects. In turn, these RNA-based therapies are likely to be more effective than traditional approaches and have a greater chance of clinical trial success and drug approval than the industry average, resulting in new medicines for patients. With almost 20 years of experience, we have honed our skills and streamlined our processes to rapidly identify and pursue new targets for potential treatment, such as the case of our drug candidate for thalassemia, via a three-step process. Firstly, our drug discovery team identifies a new therapeutic target, a disease-associated gene, and uses our proprietary algorithm powered by data and machine learning to predict the most effective siRNA molecules with the greatest potential efficacy for the selected target. Second, this siRNA molecule then undergoes a multi-step selection and optimization process involving several rounds of tests on different cell types in our laboratories. The best performing siRNA molecules are then tested for safety and efficacy using our mRNA Gold platform technology. Our platform delivers gene silencing medicines to liver cells by combining siRNA molecules with chemical address tags called GalNAcs, a naturally occurring sugar that attaches to liver cells. These RNAi molecules can then enter liver cells and silence targeted disease-associated genes expressed in the liver. This is the approach that we've taken for the development of our gene silencing candidate for people with thalassemia. So as Giles introduced, one of our primary focus areas for our research is the treatment of iron-loading anemias like thalassemia. Our candidate is an RNAi therapy designed to temporarily block a specific gene's message that would otherwise trigger an unwanted effect. In this case, the TMPRSS6 gene, which prevents the liver from producing a particular hormone, hepcidin, that controls iron levels in the body. As this hormone increases, iron levels in the blood are expected to decrease, which will in turn allow more healthy red blood cells to be produced, thereby reducing anemia. We have tested our candidate in animals to understand how it works and determined that the doses being studied in humans are expected to be safe. We reported results from our first in-man study in healthy volunteers earlier this May and are pleased to say we saw the results we wanted. In fact, they exceeded our expectations with a significant reduction in harmful blood iron levels. Now a phase 1 study is being carried out to find out more about its effects in people living with thalassemia and MDS. The aim is to find the most acceptable safe dose of our medicine with effect in people with non-transfusion dependent alpha or beta thalassemia or low-risk myelodysplastic syndrome, otherwise known as MDS, which can then be used in larger research studies in the future to understand how safe the medicine is and how well it works. The study will compare the safety and effect of our drug candidate with a placebo, a substance that is known to have no effect on the body. The study consists of 2 parts to explore different doses of our drug candidate, given as a single dose or multiple doses. It is a global study that will enroll approximately 112 participants at up to 25 trial sites, spanning Europe, the Middle East, and Southeast Asia. In both parts, those taking part will either receive our drug candidate or placebo dummy therapy. A computer decides at random who receives what. 75% will receive our drug candidate and 25% will receive placebo. Only we will know who has received what at the end of this study to be able to compare the effect of our drug candidate versus placebo. Participants and their healthcare professionals will also be notified which treatment they have received at the end of the study. If you are interested in finding out more, then please email patient-info@silence-therapeutics.com. So to conclude today's presentation, why is it worth exploring RNAi in thalassemia and our approach using GalNAc in particular? Silence's RNAi-based therapies use a very tailored and precise approach to target specific genes in specific parts of the body. This means that the effects are more potent and predictable. In turn, these RNAi-based therapies are likely to be more effective than traditional approaches. Therefore, they have a greater chance of clinical trial success and drug approval than the industry average. This means new medicines can be provided more quickly to those that can benefit from them. The targeted delivery to the right cells in the right part of the body means that most potential side effects can be anticipated. In contrast, traditional medicines are less specific as they can affect many different cell types. So off-target side effects can be more likely and challenging to predict. Furthermore, as our medicines do not cause permanent changes to DNA, any unwanted effects are only temporary.
Absolutely. And these benefits are important for patients and researchers alike, as they give the medicine the best chance of success, avoid overburdening patients with the need for frequent injections, and make sure that if the medicine leads to an unwanted effect, it is only temporary. We are conscious that a medicine is only good if it can be accessed by those that need it. So as a final point to close, I wanted to reiterate that at Silence, we are committed to working with patient organizations like TAF and regulatory authorities at each stage of our research and development to ensure that we're in the position to deliver approved medicines to those that need it across the globe as quickly as possible. Many thanks for your time, and we hope you enjoy the rest of the conference.
You go on.
So we're going to move to the last presentation of the session that will be given by Kenneth Arty. He's a pediatrician, a senior vice president and chief medical officer of the IMARA Incorporation. And he's going to present a novel agent that's called IMR 687, an investigational PDE9 inhibitor that can be used for sickle cell disease and beta thalassemia. Just to remind you that you can pose your question through the Q&A section. Dr. Arty?
Hello, my name is Ken Addy, and I'm the Chief Medical Officer for IMARA Incorporated. And it's my pleasure today to present information about IMR 687, an investigational PDE9 inhibitor for sickle cell disease and beta thalassemia. This slide is to remind everyone that IMR 687 is an investigational drug that has not been approved by regulatory agencies, and it is still in clinical trials to establish its safety and effectiveness. IMARA is a phase 2B company with clinical proof of concept for IMR 687 in sickle cell disease. We're dedicated to developing novel therapeutics for patients suffering from rare genetic hemoglobin disorders. We also have a focus on advocacy, community building, and to providing grants to support patients and families. IMR 687, also known as tabinitrin, is an oral, once-a-day, potentially disease-modifying treatment for sickle cell disease and beta thal. It's highly selective inhibitor of phosphodiesterase 9, and it's a potent small molecule, which we believe has multiple mechanisms of action through its ability to increase cyclic GNP. Our clinical program in sickle cell disease began with a phase 2A study, which is now complete in 93 subjects and showed clinical proof of concept. There's also an open label study from that phase 2A trial that's ongoing, and patients have been treated for up to 2 years now with IMR 687. In our phase 2B study, known as ARDENT, we are enrolling close to 100 patients, and this is an ongoing study, and the planned completion for that study is in the second half of 2022. Similarly, we have a phase 2B study ongoing in beta thalassemia called FORTE, which will enroll approximately 120 patients. And this is ongoing in patients both with transfusion-dependent and non-transfusion-dependent thalassemia. And likewise, the study is slated for completion in the second half of 2022. At the bottom of the slide, you'll note that IMR 687 is also being developed now for patients with heart failure with preserved ejection fraction. This slide is regarding our commitment to the sickle cell and beta thal communities. In addition to our clinical trials, we support the interconnectivity of patients with their advocacy organizations and with industry. We've been able to support community-based organizations with a grant program, and this year we were able to provide $150,000 in grants to CBOs. Sickle cell disease, as most of you know, is a multifactorial disease with a number of complications affecting millions of people worldwide. And this slide shows the main clinical findings and manifestations that need to be addressed as unmet needs in sickle cell disease. Most of these stemming from vaso-occlusive crises that result in pain, as well as acute chest syndrome and priapism, as well as these other complications shown on the slide. IMR 687 works in the nitric oxide cyclic GMP pathway, which we believe is dysregulated in sickle cell disease. And this can lead to increased inflammation and cell adhesion. You can see that PDE9 is the enzyme that breaks down cyclic GMP to GMP. So this is the enzyme that IMR 687 blocks, causing an increase in cyclic GMP, which in turn can lead to an increase in hemoglobin F, which, again, many of you know, will decrease the amount of red blood cell sickling and hemolysis, and that alone could contribute to a reduced incidence of vaso-occlusive crises. However, we think it's important to note that cyclic GMP has a role in a number of other constituents that make up these vaso-occlusive aggregates. And this includes white blood cells, platelets, endothelial cells in the blood vessels, all of which get activated and form aggregates and occlude the blood flow in blood vessels. So in a number of ways, red blood cells, as well as these other constituents in the blood vessels, indicate the multimodal mode of action for IMR 687 in reducing VOCs. So as we mentioned, the VOCs occur when sickled red cells, activated white cells, and other elements form these aggregates. Obviously, VOCs are very painful, can occur multiple times a year, and they are the leading cause of emergency room visits and hospitalizations for patients with sickle cell disease. And the painful crises can be difficult to manage. What we are attempting to do in our studies is reduce the annual rate of VOCs, which of course would decrease disease severity, improve quality of life for patients with sickle cell disease. It's also an important clinical outcome measure. If we could reduce VOCs, this would be an approvable endpoint for our clinical trials. And we've seen the potential for IMR 687 to reduce VOCs. In our preclinical studies, we can show that we're targeting red cells, adhesion, and inflammation, and that's a promising approach. In the exploratory analysis from our phase 2A study, we showed a decrease in the mean annualized rate of VOCs, as well as a prolongation of the time to the first VOC as compared with placebo. So I'd like to show some of those data to you now. This is the design for our phase 2A studies. We had a parent study which lasted up to 6 months, and patients were treated with either IMR 687 as a monotherapy, or as you can see, some patients had background hydroxyurea, 35 out of the 93 subjects. In total, we had 63 subjects treated with active drug in an escalating dose manner, and 30 patients received the placebo. After the parent study, subjects were invited to the open label extension study, what we call an OLE study. And as of this interim analysis that we did, we had 24 subjects in the OLE study. And they're receiving the higher dose here of 200 milligrams in the OLE study. And in fact, that study is now being escalated to up to 400 milligrams per day. IMR 687 is administered as a pill once daily. And so it's a very convenient medication in terms of drug administration. This OLE study is slated to continue for 4 years as a long-term safety study. This slide is to summarize the safety information and data that we received from the parent study as well as in the open label extension study. In the parent study, there were no serious adverse events or grade 3 severe adverse events reported in the IMR 687 groups. There were no clinically significant laboratory changes or changes in electrocardiograms or vital signs, notably no signs of neutropenia. In terms of the adverse events that may have led to discontinuation of the drug, the incidence in the placebo group and in the IMR 687 groups was very similar. You can see from the table the adverse events that did occur at the various dose levels in the subjects on monotherapy on the left and those also taking hydroxyurea on the right. And then the last column is for subjects that are now in the open label extension study. The most common side effects that are considered sometimes possibly related to study drug included headache and nausea. So turning to VOCs, vaso-occlusive crises were defined using traditional criteria for a VOC, and they were captured in the parent study on the adverse event form as well as on a VOC form. And you can see that the average VOC rate annualized was 40% lower in the IMR 687 treated groups compared to the placebo group. And to give you an idea what we mean by annualized VOC rates, since this was a 6-month study, if a patient had 2 VOCs in 6 months, that would be an annualized rate of 4. And that was the average rate in the placebo group, approximately 4 VOCs per year, whereas in the IMR 687 groups, it was about 2.5 VOCs per year. Now, since the data are skewed, in other words, there are a lot of patients with 0 VOCs and then some patients who have frequent VOCs, we usually use the median value when we compare the rates between 2 groups. And the median value in the IMR 687 group turned out to be 0 because more than half the patients had 0 VOCs, whereas the median value in the placebo group was 1.87 VOCs per year. In terms of the number of patients or subjects with no VOCs, it was 30% in the placebo group, but 52% in the IMR 687 pooled groups. And so this slide is showing all 93 subjects enrolled in the study. I mentioned that there was a delay in the time to the first VOC. So that's what this slide is showing. This is known as a Kaplan-Meier analysis. And we're looking at the time to first VOC as a cumulative rate on the y-axis here. So for example, if you look at the black line, which is the placebo group, it took about 3 months for half the patients to experience their first vaso-occlusive crisis. Whereas if you look at the treated groups, that rate, that time to first VOC was approximately 5.5 months. And so this was a statistically significant prolongation in the time to first VOC, the median time to first VOC. Another way of looking at VOCs is how did the patients react to the medication and to their VOCs. And so this is a patient-reported outcome measure known as Ask Me. It's a questionnaire. And 2 of the domains in this questionnaire concerns pain episode frequency and pain episode severity. So we're showing you the change in the score over time from the beginning of the parent study to week 24 and broke it out by the patient groups. And what you can see here is that in particular, for the pain episode severity, there was a lower score in the high dose group compared to placebo, and lower scores indicate improvement on this scale. And this was statistically significant for the high dose group versus placebo. So we feel this is an important way of seeing what the patients feel by using patient-reported outcome measures such as this. Finally, we are interested to figure out what of the many mechanisms of action may be driving some of this improvement that we're seeing in VOCs. And one of them could be that we're increasing the amount of fetal hemoglobin. That could be measured in 2 ways. One is by looking at the percentage of hemoglobin that's hemoglobin F, or fetal hemoglobin, that's shown on the left. And on the right is the percentage of cells, red blood cells, that have a substantial amount of hemoglobin F in them. And what we're showing is the absolute change. So in other words, if the patient had 10% hemoglobin F, this would be an increase of to 15%, for example. And same with the F cells, we're showing the absolute change in the percentage of red cells that have hemoglobin F. And each line here is an individual patient. So in this waterfall plot, you can see the majority of patients increased their hemoglobin F, and a majority of patients increased the number of F cells. It's about 1/3 of the patients having at least a 3% absolute increase in hemoglobin F, and about half the patients having an increase in F cells, absolute increase of 6% or greater. So based on these results, we initiated a phase 2b study, the ARDENT study. It's a global randomized double-blind placebo-controlled trial of IMR 687 in adult patients with sickle cell anemia. This study is fully enrolled. You can see the eligibility criteria here, and the endpoints for this study are quite similar to the ones we were just talking about: hemoglobin F, rate of VOCs, and time to first VOC, among others. So we will have an interim analysis of these data coming up over the next few months, and the final analysis will be in the second half of 2022. This study is testing placebo versus 2 dose levels of IMR 687, again, given once a day by mouth. And again, I'll just emphasize that this is a global study, and you can see the countries that are participating in our sickle cell disease phase 2b trial. Turning now to beta thalassemia, again, this is a global disease in terms of the number of patients affected in regions around the world. And the symptoms and signs of this disease depend on whether patients are being treated with chronic transfusions, what we call the TDT cohort, or those who are not transfusion dependent, or NTDT. And the complications shown on this slide, many of you are familiar with, are largely as a result of severe iron overload due to the transfusion. So the goal there is to reduce transfusions if possible. On the right, you can see that there are a number of complications that are related to chronic anemia, ineffective erythropoiesis, as well as iron overload, and they are shown on the right. Excuse me. So the mechanism of action in beta thalassemia is similar. We're still trying to increase cyclic GMP, which we feel should drive an increase in hemoglobin F. Cyclic GMP also has antioxidant properties. So there's perhaps a number of ways in which we can reduce ineffective erythropoiesis, get more healthy red blood cells out of the bone marrow, leading to an increase in hemoglobin, decrease in transfusion burden. The preclinical data for beta thal are shown here and will be presented at ASH later this year. These studies use an experimental mouse model for beta thalassemia, and the animals were treated with either 30 or 60 milligrams per kilogram per day for 30 days. So this is similar to the human equivalent doses that we're using in the clinical trials. And you can see on the left an increase in hemoglobin levels in a dose-dependent manner. And we also had an increase in red blood cell count and reticulocyte count. And then on the left, you could see an improvement in terms of the maturation of the red blood cells. So there's a decrease here in the ratio of immature to mature red blood cells. This is in the bone marrow, erythroblasts. So these data suggest that IMR 687 may be an effective therapy for the treatment of beta thalassemia. We have the phase 2b study ongoing, known as Forte. It is almost fully enrolled. And again, this study will read out at the end of 2022, and we have interim analyses along the way, the same dose groups as in the sickle cell disease trial. It's primarily a safety study. The secondary endpoints include efficacy in terms of reduced transfusion burden and improved hemoglobin and hemoglobin F. So in summary, IMR 687, or terbinafine, is a highly selective inhibitor of PDE9, and it's a potent small molecule that should increase cyclic GMP, and as such, could be a therapy to provide benefits to patients with sickle cell disease and beta thalassemia. In sickle cell, we could see an increase in fetal hemoglobin, decrease adhesion, decrease inflammation, and improved blood flow. In beta thal, we might see increases again in fetal hemoglobin and improved red blood cell maturation. It's an investigational agent that's given as a pill once daily. In that sense, it's easy to transport and to store and to administer to patients. And for that reason, we've embarked on global studies with our drug. In the phase 2A study in sickle cell disease, it was generally well tolerated in terms of safety, both as a monotherapy and in combination with hydroxyurea, with no treatment-related serious adverse events. And exploratory analyses show that IMR 687 could lead to a reduction in vaso-occlusive crisis. In the beta thal mouse model, we're seeing improvements in hemoglobin and red cells. And we encourage you to follow the Imara clinical trials. Here are the locator numbers in clinicaltrials.gov, as well as the contact information for Imara. I'd like to thank everyone very much, and I look forward to your questions.
I would like to thank all the speakers for their excellent presentation. Now have plenty of time for some discussion, to answer some of the questions that have already been posed in the Q&A session. So if there are some more questions, please do not hesitate to write your question and we'll respond it. So let's go directly to what we have so far, and just give me 1 second. There is a question for Dr. Eleftheriou from Dr. Arabdani from Mauritius. Dr. Eleftheriou, talked in her presentation about the use of hemoglobin F inducer in sickle cell anemia. Can these products be used in thalassemia patients as well? Or are the trials including thalassemia, or are these trials including also thalassemia patients? Perla?
Yes, lovely. Thank you very much. Yes, and I can say that, yes, there is a promising message here. Absolutely. I mean, induction of fetal hemoglobin has been our interest in thalassemia for a long time. And as we just heard, actually, from Kenneth, there is a promising trial going on now in thalassemia, which is a randomized, placebo-controlled phase 2B trial, which evaluates the safety and tolerability of the IMR 687 phosphodiesterase 9 inhibitor, looking at the effectiveness and safety in 120 adult beta thalassemia patients, transfusion dependent and non-transfusion dependent thalassemia patients. So yes, there is a great interest, and there are clinical trials looking at this going on.
Great. There's one more question. I guess that will be probably for Stefano. Is the treatment, this is from Mrs. Mutoyani, could treatment with luspatercept affect negatively a possible future gene therapy?
Can you hear me?
Yes, we can hear you,
Okay,
Stefano.
excellent. Okay, very good. Well, thank you. So first of all, thank you so much for inviting me here today. So I would actually defer maybe this question to Dr. Cappellini and other clinicians that are actually using luspatercept right now. My personal feeling is that this drug should not prevent patients eventually to decide to go for a gene therapy trial. So that would be my immediate response, a gut feeling response. But I rather ask Dr. Cappellini maybe to provide an additional comment to this question.
Thank you, Stefano. It's a very reasonable question, and my answer is similar to that of Dr. Rivella. I don't see any preclusion to move to gene therapy after luspatercept treatment. And I have to say that this is actually the attitude of some patients today. They want to start being treated with luspatercept and in case of no response to consider gene therapy. in the past was the opposite. Gene therapy was seen as the dream and the first choice. Now is coming second after luspatercept. I don't see any reason to avoid treatment of luspatercept before moving to gene therapy. Just is this, I think, and the guess. I would say that in some cases of gene therapy uncompletely successful, the introduction of luspatercept could be helpful and improving the outcome of gene therapy.
Right. So, Nika, as I alluded in my slide, you probably can address this better than I do, but I wonder if the use of this drug or even other drugs, right, may actually decrease the excess of ineffective erythropoiesis and erythroprogenitors that we know can be a limitation to myeloablation and selection of CD34 cells. So do you see an advantage actually using this kind of treatments as a pre-treatment for gene therapy?
Yeah, it could be. We don't have proof of that, but in theory is very, very real and possible. And considering that the myeloablation remain one of the main challenge of bone marrow transplantation, gene therapy, and so on, I believe that it could be a pre-gene therapy treatment in some way. But, you know, Stefano, now is a theory. We have to prove it. And I hope that since the drug will be in real life, and it will be soon in several countries, we may design such independent study.
Yeah, and I think that, Stefano, you are the best person to prove whether this also it's something that it actually happens or not. So if you think that an experiment about whether CD34 uncommitted cells increase with luspatercept, I think that would be an excellent thing to do, an excellent experiment to do.
Yeah.
I
We have a
could
couple more.
that Antonio can add here. Having worked at Acceleron for many years on luspatercept, we do know that within about 2 months that the hemoglobin levels will start to come down again, perhaps to baseline levels. But that's in some ways a good thing, right? So you have a therapy that's reversible, and so if you decide to go on to another treatment, I agree with the other speakers that I don't think there should be a problem. Probably the question is arising from, if you do one gene therapy, does that preclude another gene therapy? I think that's a realistic question.
Yeah, that's a very good point.
Yeah.
We'll have a couple more questions from the audience. Again, from Dr. Arabdani for Dr. Rivella. If you can please elaborate a little bit more on the non-toxic myeloablation, because any alternatives or development or trial, as most of the patients having transplant or gene therapy face significant complications due to infection and immunosuppression.
Absolutely.
Stefano?
I think that from my perspective, my vision, the next 2 steps in the field of gene therapy would be one step where we may be able to identify drugs that are more selective for hematopoietic stem cells. So there's been an attempt in the last few years to generate antibodies specifically targeting hematopoietic stem cells. I have to say that so far this approach didn't translate in an effective reduced myeloablation regimen, right? Unfortunately, I think, however, that the studies are there are studies and there are new probably modification of this approach that eventually may be available in the future. So just want to clarify, this is still a myeloablative regimen because obviously you're targeting the hematopoietic stem cells. But the idea is to be more selective and, for instance, not to target germinal cells, not to target the endothelial cells in many organs, or the gut, enterocytes, etc. So from that perspective, this drug could be more selective and less toxic. I think the next step would be complete elimination of myeloablation, but that would, it means, as I alluded in my presentation, to develop therapies that target directly hematopoietic stem cell in vivo. And I have to say, in my lab and other laboratories, I know there are a lot of studies going on right now to modify lentiviral vectors or use RNA-based sequences to really, and let's say microvesicles or LNP particles, to target directly these genetic elements or genetic modifying elements directly to the hematopoietic stem cells. So that would be something that would eliminate completely the myeloablation. Again, I'm not talking something that is around the corner, unfortunately, so I don't want to raise any expectation that these techniques would be and would be available in the short term. But I think a lot of people are working in that direction because we believe that this would be a major step forward in the treatment of the patient and hopefully also decrease the cost.
Yeah. Very well. We have a couple more questions. I have one from Mrs. Maria Hadjidimitriou, and again, it's for Stefano. With Bluebird Bio shutting down the European Gene Therapy Division, what are the options for any curative therapies for our European patients? So I guess none at this point.
Right. And I'm asking everybody to contribute here because I'm probably not the only one that can provide some elements to this question. So I have to say that I'm personally disappointed by the decision by Bluebird. And I be very honest and frank, I think it has been an economical decision in order not to decrease the cost of the therapy in the United States. Because if you you can offer at a lower price a product in Europe, you may need get pressure to lower the price in other countries, right, where you can sell the same product for a higher price. So I think I hope that the Vertex product eventually will be available in Europe. That should be easier to provide, also less expensive. Personally, I'm actually very interested in delivering our new product in a way. We're trying to really seeking, I can't say that we already have, but I'm seeking strong partnership that allows us to make the vector a much lower price. I have to say that some cost is required at the beginning. My philosophy is that we need to show that this is successful and safe. But if this is going to be efficacious, I really hope that the competition will lead us in the sense, developing more and more a better technology that would be cheaper and available to everybody. Even if, if the cost is going to be lower in Europe and hopefully also in the United States, it may still be very high for other countries. And so we really need to work hard to make these available to everybody. So I'm sorry for the long answer with a limited response in a way, but I think many people share my thought about this, and I'm sure that other people can contribute to answering this important question.
Yeah.
Yeah. Well, sorry, if I can make a comment. Of course, this is an issue. Because beside any company decision which is out of our control, and we cannot, of course, interfere on that, but in such a situation, it generates inequality. At this very moment, European patients are in a disadvantaged position compared to USA patients. So I think we have to work, as Stefano said, in this direction to find a way to guarantee or to offer the same option to all the patients. So companies, they can make their choice, but we as scientists have to work in order to have let's say, equality in treatment. That's what we want. And for sure, we will be with Stefano helping in trying to develop other procedure or other vector, whatever you like, who could be more affordable.
Yeah, I think that's the example that was said by Bluebird Bio and the whole story of Bluebird Bio and the products in Europe. It makes actually be a good example for the other companies not to have a very high expectation and to have products that can be affordable from the healthcare systems. Otherwise, they will not be able to. The system will not be able to afford that. So unfortunately, this was a very unfortunate twist of events, but it's also a good lesson for everybody. I think the company. Yeah.
Can I just quickly
Pardon.
add something? I agree with everything that has been said about it, but I also want to stress the importance of working as hard as we can with the authorities as well, both in the UK and in the Europe, and just push this for our patients, because at the moment this is thalassemia and sickle cell disease are prioritized very much lower than anything else, and advocating for our patients and for all the treatments that are available, regardless of the costs, it's really of paramount significance, and it's our duty of care for these patients. It's about, regardless of the costs, really, although, I share what you just said about, it's a big call here for the companies to be more approachable, really, to make treatments more affordable for these patients, but it's also our duty of care to the patients to ensure that, we convince our authorities to reach their hands in the pockets and get out, what they need to get out in terms of financial support to provide equity of care to all these patients.
I definitely agree with that. Everybody agrees with that, but also it's our responsibility to persuade the pharmaceutical companies that they need to decrease their costs.
Yes.
With the same way that
Definitely.
we need to ask the health authorities to accept it. Let's go to a couple more questions, and I believe that we have the permission to use 5 minutes from the break. There is one question by Dr. Cheng. Do you know that factors deciding which patient respond with hemoglobin F in the IMR 687 study? And if there's a way to predict which patient will best benefit from that? This is for Dr. Ratti.
Thank you for that question. I don't think we were able to define in the phase 2A study which patients were more likely to respond, because I think we did have patients having a reduced, for example, in the sickle cell trial, reduced VOCs, vaso-occlusive crises, who didn't necessarily have a dramatic increase in hemoglobin F. So we are sort of beginning to step back and say that is one of the mechanisms that the drug is working, but there are several others. We talked about white cell adhesion, the vascular endothelial cells, even smooth muscle cells and calcium efflux, even vasodilation, because phosphodiesterase inhibitors have a vasodilatory effect. So if you take all of these different contributing factors, then the hemoglobin F becomes less critical to predicting whether or not we'll see a reduction in the vaso-occlusive crises. And even in thalassemia, the anti-inflammatory, antioxidant properties could have a number of benefits in beta thal, not all of them being measured by hemoglobin F or total hemoglobin.
This is very similar in sickle cell disease as well. You see some patients who don't really respond in terms of the fetal hemoglobin very well, but yet, they have significant clinical response.
And I have one comment and actually a question for, I guess, for both Kenneth and Dr. Campion and Martinez, whether you think that providing studies for TDT and TDT together can render more difficult to extrapolate results, and who is going to be the patient that will respond better to the study drug. And we have seen that in very many studies recently. So your opinion, please. Kenneth, first.
Well, I would say it's fairly important to do both at the same time, because if you think about sort of the chain of events, in order to see transfusion burden reduction in a thalassemia transfusion-dependent patient, you really, the physician has to see the increase in pre-transfusion hemoglobin, react to it, decide whether it's appropriate for that patient to either increase the interval between transfusions or reduce the number of units per transfusion event. And that's sort of a sequential, that's the subjective part of transfusion burden, whereas hemoglobin, hemoglobin F, some of these other things are more objective. But we can't really rely on those kind of data from the transfused patients because the transfusions interfere with our interpretation of those tests. So I like to look at thalassemia trials using both. We did that in our phase 2 trials for luspatercept. We had the arm with TDT, the arm with NTDT. You're looking at the hemoglobin response here. You're looking at the transfusion.
Yeah.
Okay, Dr. Martinez.
Yeah, no, I agree with that.
Okay.
Good.
And I guess, do you want to have, okay, I think that there's the last final question. I think for Stefano, you said something about a microneedle. Do you know if this was going to be used for Desferal or other injectable iron chelators? Or
Yeah,
this
so
only has...
I think this is a good question. I'm trying to submit the information about the people doing the clinical trial for this device. I suspect that could be utilized for reducing pain for injection in general. So I don't have a direct answer for that, but I'm trying to provide in the chat information about the location where the group is doing the clinical trial, and so maybe the patient can reach them out directly and ask these questions. And maybe if they didn't think about it, then maybe it would be a good approach, a good application for this device as well.
Lovely.
Okay.
And I think we need to close the session. I would like to thank you all for your attendance and all the presenters for their excellent presentation. Nika, do you want to close?
Yes, I also want to thank the speakers, but all the attendees and those who sent questions. So hopefully we may enjoy the rest of the meeting, and we are going to close this session and having a short break. Thank you.
Thank you all.
Thank you.
Thank you.